So here we are now with Professor Justin Kumar of the Department of Biology here at IU, and he's going to tell us a little bit about his model system he uses for his research today. So Justin, the first question I want to ask you is, what is it that got you enthused and excited and interested basically in the model system you use and the questions you ask?
Well, we use Drosophila, as you know, and the great thing about Drosophila is that it is a very simple system. And I study the eye specifically, and it's even simpler than the fly as a total. It has several hundred repeating units. Each unit has exactly 20 cells, and each unit develops just like the others. So it is really almost like a crystal. It's a neurocrystalline lattice that one can look at and study.
That's pretty cool. So regarding at least even eye development and structure, I mean, how similar is that to the human eye, and is it relevant in research of the human eye?
Well, obviously it looks very different. If you look at a compound eye, look at a simple eye, you would assume that there are no similarities between the two. And that's what people thought of for more than 100 years when people studied these sorts of issues. But as molecular biology has been used to clone genes and try to understand what creates a compound eye, what creates a simple eye like we have, all the genes have been shown to be the same. So anything that you study in a fly eye can actually be used to study eye development in human eyes, in any eye of a mammal, even eyes for fish and octopi, and so on and so forth.
Okay, that's pretty cool. So, I mean, obviously you have that strong interest in terms of eye development and function. What are the strengths of this model system? The fruit fly, so what's the genus species again?
This is Drosophila melanogaster. And the strengths of the organism is that it's small, that you can house hundreds of different mutants in small vials, put them in incubators. You can actually grow millions of them at a time. They have a very quick generation time, about 2 weeks. So it's unlike mice or unlike primates in which you need very large facilities. Here you can have a small incubator that can house millions of flies in a small space. Other things that make it great are that you can do genetics, you can create mutants, you can actually look at the phenotype, what happens to the physical body of the animal, and study anything that you're interested in, whether it's eye development, whether it is pigmentation, whether it is limb development, whether it is learning and memory. There's all sorts of tests and assays one can do for any trait that you're interested in.
So physiology, even all the way up to behavior.
Absolutely. People use the fly to study pain sensation, learning and memory, taste perception, flight, walking up and down, geotaxis, any sort of behavior one can study with them.
So in that regard, we had Dan Kearns tell us some of the strengths of his system, and we're curious, in addition to strengths, also weaknesses. So do you feel like there are any weaknesses regarding the fruit fly system and questions that you might not be able to ask as well as other systems?
Well, I hate to admit it, but there are at least a couple of ones that I can think of. One of them is that it's not a good biochemical system. You cannot get large quantities of proteins from an individual tissue. The fly is just too small. So if you're looking for something big, sea urchins, organisms like that are much better for biochemistry. The other thing is, if you're looking at cell biology, looking at the cell cycle, looking at chromosomes replicating and dividing, flies are also not good for that. The cells are just too small. What you really want are nice fibroblasts that are grown in on a petri dish, big cells that you can really look at. So, as I said, I hate to admit it, at least there's two things that I can think of that aren't so great. But for anything genetic, anything developmental, anything molecular, flies are one of the premier systems.
So in that sense, laboratories will utilize multiple model systems for research, but also for practicality purposes, expressing proteins and doing different biochemical things. Are there any other model systems you utilize in your laboratory?
Well, certainly for any routine cloning, we use bacteria. Because once we clone a fragment into a plasmid, we have to actually multiply it and grow it up in large quantities, so we use bacteria. So in that sense, we use it for model system. We have collaborations with Richard Hardy, who's a virologist in the department. He studies viruses, and so we're looking at how viruses and flies can interact and how viruses take advantage of host factors in the fly to actually aid in their replication. One can also consider that we're going to look at mice and zebrafish, sort of vertebrate systems later on in the future, and so those will get incorporated into the laboratory. Use what we know in flies to try to understand higher development.
Excellent. Well, this has been Professor Justin Kumar, professor in the Department of Biology here at Model System Drosophila melanogaster. We learned a lot about physiology and the basis for doing genetics in Drosophila. Thanks, Justin.
You're welcome.
Appreciate it.